Good morning, children. So after the completion of the second chapter, today let us discuss the few questions which is related to the NEET. That is nothing but competitive. Okay? Yeah. So while we are discussing these questions, all of you should have at least pen or pencil with your hands and note down some points, because while we are discussing you may get a number of doubts. So note down those doubts also. After that one, you just send me the messages. If you're having any still doubts, means let me to clear. That's why. Okay? Yeah. Let's start the discussion. The first question. So this question, it is related to the topic pre-fertilization, structure and events. So that is nothing but structure of a flower. Okay? Yeah. So this topic is nothing but the logic distractor, or we can also say exactly it is related to the particular which topic. Okay. The first question. Analyze the statements given below. Okay. So there are three statements they have given in the form of P, Q and R. The first statement, read properly. The several hormonal and structural changes lead to the differentiation and further development of the floral primordium. Second statement, that is Q, inflorescences are formed which bear the floral buds and then the flowers. The third statement, R, in the flower the male and female reproductive structures, the antecium and the gynoecium differentiate, that means develops. Okay, so in that one, see, if you see all these statements, these three statements are all correct. Yes, because this topic we have discussed during the when it comes to the during developmental stages, vegetative stages, and during the developmental stages, because of the hormonal changes, what are the changes will happen. So those all things we have discussed in the first question. I think it's clear. Okay, got it? Yeah. So next question, second question. So here the diagram given, a diagrammatic representation of longitudinal section of the flower is given, isn't it? We have to label it. So without going towards the whatever the given options or the answers, just you recall the structure of the longitudinal section of the flower so that you can identify. For example, A, that is nothing but filament, isn't it? The B is nothing but colorful structure, petal. C, that is nothing but anther. And the D, yes, stigma. What about E? Style. And F, yes, that is nothing but disc. And G is nothing but ovary. H is ovule. And I is the sepal. So it's clear that the labels, okay, the labels of the given diagram of LS of the flower. So B is the right answer, isn't it? Yeah. So this question is related to a diagrammatic representation of LS of a flower or typical flower. I think it's clear. Yeah. Next question. The next question, it is related to again a typical stamen. Okay, the typical stamen, it is given, isn't it? A typical stamen and a three-dimensional cut section of an anther is given. You should identify the again labels, isn't it? Yeah, as I said, without going towards the whatever the options, you can just label it so that you will get easily. The first label is a pipette anther. The second label indicates the pollen rind. The third label is pollen sacs. Fourth label is the line of dehiscence, or we can also say a split open. The fifth level is nothing but filament. The right option is yes, B is the right option. I hope you can understand in the easiest way. Okay, yeah, fine. Because sometimes without labeling, if you move towards the whatever the given, the options, it leads to a lot of confusion. So instead of that one, if you label, so after that you just compare the whatever the given options. It will be easy. Okay, yeah, fine. The next question, the fourth one. A typical angiosperm anther is, you know very well that while we are discussing about the atypical, the angiospermic anther, we told you it is nothing but, yes, bilobed. We also told that a terminal portion is generally has bilobed club-shaped structure called anther, and the bile is nothing but two lobes, and also we said diatheca, that is they are two branch-like structures, or we can also say around-like structures, bilobe and diathecus. I hope you understood easily. Whatever we have discussed earlier, you just recall that one, you will get it easily, isn't it? Yeah. Next question, the fifth one. Correct sequence of layers of anther from outer to inner, and sometimes in examination they may ask from inner to outer, like that also they can ask. So write down the logic distractor for this one. This question is related to the TS of a young anther. I hope you remember where we are discussing about the structure of microsporangium. We have discussed about. Okay, if you see the diagram, easily you will identify. Yeah, outermost region is epidermis, then after endothelium, after that the mid layer, inside the innermost layer, that is tapetum, nourishes the developing pollen glands, right? Yeah. So this is what we can identify. You just observe the diagram. Okay. So sometimes they may ask from inner to outer, means that will be vice versa. Okay. Beginning from tapetum, mid layer, endothelium, and epidermis. Like that, you can identify. Got it? Yeah. Next question, the sixth one. Select the true statements regarding tapetum. See, while we are selecting these statements, we should select only true, but regarding tapetum. That means these are all the statements, whatever they have given, we should identify regarding only tapetum, not related to any another layer. Okay? Yeah. So here there are five the statements. Innermost wall layer, then it is a nourishing tissue, and then third, cells possess dense cytoplasm. The fourth one, cells are generally having more than one nucleus, that is many multinucleus. Then the cells are wedge shaped. Yeah. So in the diagram you can identify again this question. It is related to the transverse section of the young anther. Specifically, these statements are related to the tapetum. Almost all the statements are true. That is nothing but correct. So that the option is E. All are correct. Okay? Yeah. So then next question, the seventh one. Microsporogenesis means, here we should identify just the definition. What is the meaning of microsporogenesis? What is the microsporogenesis? How can you define? The formation of microspores from pollen mother cell, that is PMC, through meiosis is known as microsporogenesis. I hope you can remember while we are discussing about the topic. I told you clearly how it will be happens. You can also say the process of formation of pollen rods or microspores from pollen mother cells through meiosis is called microsporogenesis, isn't it? Yeah, so option C is the right answer. The formation of microspores from pollen mother cell, PMC, through meiosis. Okay? Yeah. Next question. Eighth one. Okay? The male, that is nothing but male gametophyte in angiosperms, starts with. So that is nothing but, it's easy question, we can easily understand. The pollen runs are nothing but male gamete. Okay? Yes. The pollen lines represent the male gamete. I hope you can remember the structure of the mature pollen lines while we were discussing. I told you the pollen lines represent the male gamete. That is but we can also say male gametophyte, isn't it? The pollen lines are generally spherical and they are measuring about 25 to 50 micrometer in diameter. These are all things we have discussed, isn't it? Yeah, fine. So that the option B is the right answer. It's easy question. The next one. Yes. So here, as we discussed earlier, the structure is given. So that based on these tables, you should identify the whatever they have given, isn't it? The enlarged view of one, that is microsporangium with wall layers are given or shown. Identify the layers. Okay. Yeah, here it's also sometimes, see here it is given, regularly. To confuse, sometimes they will not give regularly. Irregularly, they will jumble the labels also. But while you are identifying, so as I said, it's an easy method. Just write down the labels, then after compare the labels with respect to the option, so that there will be no confusions. Got it? Yeah. See here, the first layer, outermost layer is epidermis, isn't it? Then the next layer, the second layer is nothing but endothecium. Then after, we can identify the mid layer. Then the fourth one, what they are given, megaspore. Okay. Yeah. So here, while we are discussing about the microsporogenesis, MMC means microspore. Okay. Yeah, because while we are discussing about microsporogenesis, megasporogenesis, it will diffuse. Here it is MMC means microspore mother cell. Then the fifth label, it's a tapetum. Got it? Now just compare these labels with respect to the options. Yes, option B is the right answer, isn't it? Yes, it's correct. Yeah. The next question. Regarding the pollen runs, that is nothing but male gametophyte. Here it's also we should identify what are the statements they are given, how many of the statements are correct, or we can also say true or false, like that they will ask. Okay, let me to read the statements. The first statement, generally bilobed in shape. Then the second one, the size may be 25 to 50 micrometer in diameter. Then the third one, its outer covering is made up of pectin. Then the fourth one, exine has germ pores where sporopollenin is absent. Okay. See here, almost all, okay, though generally bilobed in shape and its outer cover is made up of pectin, it is not, always true, because the bilobed also, I told you while we were discussing in our subject and whatever in our syllabus they have given the bilobed. Sometimes it may be mono or sometimes it will be tri, it will be diffused. But the second statement and the fourth statements comparatively to the pollen lines, it will be always correct. Okay, the size maybe varies from one species to another species. It is 25 to 50 micrometer. Then the exine has a germ pores where the sporopollenin is absent. Got it? Why the third statement is false? Because outer covering is made up of sporopollenin, not pectin. Inner layer, that is inner covering is made up of pectin. Okay, that's it. Option A is the right answer. I think it's clear. Yeah. The next question, the 11th one. This is also the question related to the pre-fertilization structure and the events. Yeah. Intine is made up of the composition of the intine. Okay. So while we are discussing about the structure of the mature pollen rind, I told you it has namely two wall layers, the outer exine and inner intine, right? Yeah. So outer exine is made up of sporopollenin, isn't it? What about the intine? The intine is made up of cellulose and pectin. Okay, the intine is a thin and a continuous layer made up of cellulose and pectin. That is the composition of the intine. Got it? Yeah, this question is related to the composition of the a layer of the pollen rind. The next question. One of the statements given below is not suitable for the sporopollenin. See, this is vice versa. They are given. We have to select the statement. It is not suitable. Okay. So it is just like not suitable or suitable. Like that, they can ask the questions. Let me to read the question. You can read all the statements also. Exine is made up of organic material. Then the D, it can withstand high temperature, strong acids and alkali. Next, C, pollen rinds are preserved as a fossils due to the presence of sporopollenin. Then the fourth statement, it is easily degraded by most of the enzymes. False. See, whatever they have given three statements, A, B, C, these statements are suitable. Okay, this suitable, that means correct with respect to the sporopollenin, isn't it? But the D, what they have given, it's a false. It is easily degraded by, they have given, but it's a false. It is not easily degraded by, isn't it? I told you the exine is made up of sporopollenin, which is one of the most resistant organic material. It can withstand strong acid and alkali. No enzyme that degrades sporopollenin is so far known as. Because of this property only, the pollen rinds can be preserved as a fossils. I hope you can remember, isn't it? Yeah. The next question. So did you got it? Yeah. Next question. The vegetative cell of pollen rind is, okay, the vegetative cell of the pollen rinds. So there are two cells, isn't it? In pollen rind we can identify two cells. One is the vegetative cell, another one is generative cell. So what they are asking the question, the vegetative cell of the pollen rind is having abundant food, that is reserves. Okay, because of this property only, it provides nourishment, okay, during further process. So option B is the right answer. Okay, yeah. Why the remaining statements are false? You can easily identify. They are given smaller than generative cells. No, vegetative cell are always bigger than the generative cell. The generative cell is smaller than, isn't it? Yeah. And they are given spindle shape with dense atom. No, it is not a spindle shape, isn't it? Then with a small spherical nucleus, no, vegetative cell which is having, that means greater or larger nucleus comparatively to the generative cell. Got it? So that option B is the right answer. Next question. Question number 14. Read the following statements regarding pollen rinds. Here it's also, this question is related to the pollen rinds. Okay. Yeah, let me to read the statements. Yes, you just identified. They are rich in nutrients. Yes, it's correct. The pollen rinds of many species such Parthenium or carrot grass cause severe allergies, often leads to chronic respiratory disorders. Yes, this statement also correct. The third statement, it is better to use immediately because they cannot be stored for further purpose. Some species it is not applicable, but of course. Then pollen tablets and syrups are used to increase performance of the athletes and race horses. Yes, correct. So whatever they have given, 4 statements regarding 14%, it is related to the pollen grains, all are correct. So which is the right option? Yes, option D is the right answer, because all the statements 1, 2, okay, and 4. Because why the third one is not correct, I told no. So it is better to use immediately what they are given. I told you, because of this sporopollen, it can be stored, or it can be withstand for longer duration. But so this statement is not suitable for universally acceptable. That third statement is only suitable for particular species. So that the option D, first statement, second statement, and the fourth are correct. Okay, yeah. Next question. It's a 15th one. Viability of pollen grains of some cereals such as rice, wheat, etc. is. Viability means the ability to survive until it reaches to the surface of the stigma or stigmatic surfaces of the female reproductive structure or granulation. Okay, it is direct question. In they are mentioned also the cereals such as rice and wheat is about only 30 minutes. Okay, what will happen if that time is exists? They lose the viability. Okay, they will lose the ability to survive. Okay, so that's why the viability of pollen grains of the some cereals such as rice and wheat, it is about only 30 minutes. Okay, yeah, but it will be diffused. I told you, when it comes to some species, they can survive for years together, and sometimes, okay, in the last topic when we are discussing, the seeds are survived. Okay, they are also found as fossils. Okay, yeah. The next question. Observe the diagram. The diagrammatic wave of a typical ovule, it is also known as megasporangium, is shown. Select the correct labeling. Yeah, here also, before you are going towards the option, just label. Okay, just label it. You can identify easily. Yeah, A label is hilum, right? Yeah. Then B, funicle. C is a micropyle region. The D, micropylar pole, that is micropylar pole region. We can also say micropylar end. Then E is the outer integument. Then F is the inner integument. What about G? Yes, G is the nucellus, which is a nutritive tissue. H is the embryo shed, and I is the chalazal pole. Now it's clear. The option B is the right answer, so that it will easily, okay, you can identify the diagrams with respect to labeling. Got it? Yeah. So which is the right answer? Option B. Next question. So this question, see the match the following they have given. What is this related to? That means option, what are they given, the A side and the B side. A, they are nothing but the types of the pistils. Okay. Then B, what they are given, that is just matching towards the types or with respect to the, so what is the opposite. So for example, let me to explain. Option A, in the first one they have given it is a single pistil. The single means one, that is mono. Okay, type of the pistils, monocarpellary. So first one is E. Then second one, fused. while we are discussing about the types, I told no, apocarpus and syncarpus. So here the fused means, yes, syncarpus. Apo means free, syncarpus means fused. Then the third one is stigma. It's very well known, the landing surface or landing platform of the gynoecium. Then the ovarian cavity, yes, they are nothing but locule, a room or chamber-like structures which is present inside the ovary. Then the fifth one, megasporangium. Megasporangium, they are commonly known as ovule. Okay, I hope you can remember when we are discussing the structure of anatropous ovule. We said that megasporangium. Then the sixth one, integments. It's a protective envelopes. There are two integments which is present, the outer and inner. So which is the right answer now? Option B is the right answer. Okay? Yes. The next question. Over 60% of angiosperms shed their pollen rings at. Well, we are discussing about the pollen rings, I told you. So, the pollen rind generally, whatever we have discussed in our, the syllabus, it is two-cell stage, isn't it? That means it containing one vegetative cell and another one is the generative cell. That means over 60% out of 100% angiosperms, or only 60 or 60% of angiosperms releases or shed their pollen rings at two-cell stage. What about remaining? There are possibilities of the three-celled stages also. I told you there are some specific species may shed. In such a cases, the vegetative cell may be single cell, the generative cells may be two cells. Okay, but here whatever we have discussed in our syllabus, they are given two-cell stage. Then which is the right answer? Option A is the right answer. Next question. This one very easy. Here it's also, instead of going towards the whatever they are given the answers, just label it. Labels so that you can easily compare the after whatever they have given the options. A, yes, it is antipodals. B, that is polar nuclei. Then C, central cell, large central cell, we can also say. And the D, egg, right? Yes. E, synergids. What about F? They are nothing but filiform apparatus. Now just compare this with respect to the option. Which is the right option? Yes. Option B is the right option. Okay, easy to understand. So that you can also easily identify the option, right option. So next question. Here, add one. Okay, add ones which is just like which is not suitable or selectable with respect to particular group. Okay, add one out based on number of ovules. See, they are given. Here you can easily, there is no need to in-depth knowledge. You just identify the wheat, paddy and mango seeds, okay, and papaya, isn't it? The papaya may produces huge or many number of the, that is, ovules, isn't it? But wheat, paddy and mango, comparatively the wheat will be monocotyledon, paddy is also, of course mango is dicotyledon, but it will be one fruit will be containing only one ovule, isn't it? But what about in case of the papaya? They are nothing but many, ovules are many. So that's why the papaya is an odd one, okay, based on number of the ovules. Okay,